Yeah, so hello and welcome everyone to the varvis workshop here at VPTC. We're happy to be part of the conference again. We did this a couple of years ago back in Prague, but this time in a virtual setting. It's a bit different. So we're here today to present to you the varvis software to show you how to use it, and especially with the use case of whole exome analysis in clinical diagnostics. So can I? Yes, I can. So this session is basically divided in 3 parts. We will start with a software demo after the introduction, where my colleague Irene will show you the software in detail and some nice use cases in the software. We then have an introduction to the additional services we're offering, like training programs, support, onboarding services, validation, and so on, also presented by Irene. And finally, we have Lena on board, who's like the head of service at Limbus here, and she will present some very interesting, extraordinary variants and how you can detect and interpret them with the help of varvis. So before we start, let me briefly introduce to you who we are. My name is Yvonne Kassmann. I'm the head of marketing and sales here at Limbus, and I'm also responsible together with my team for customer training and onboarding. And varvis is developed by a company called Limbus Medical Technologies, and we are located in the north of Germany, in Rostock, right at the Baltic Sea. As you can hear in my voice, where the flu and cold season has already started, so I don't usually sound like this. Limbus is a software development company and medical device manufacturer, which means that we operate and manufacture our products according to international quality standards. So varvis is a cloud-based clinical decision support system and a CE-labeled medical device, which is also what distinguishes us to many other products or platforms that you have seen earlier also in the course. So it's not a research use only product, but really designed to aid in the diagnosis of patients. The varvis genomics platform is like a turnkey solution for all clinical NGS applications. So you can use it to analyze targeted NGS panels of all sizes, whole exome sequencing, even whole genome sequencing. So apart from the germline analysis, we also offer somatic solutions, so support cancer diagnostics, even for high sensitivity assays, the so-called NGS-based liquid biopsy. We also have a solution for the SARS-CoV-2 surveillance program, which we hope will come to an end soon. The varvis genomics platform basically consists of 3 parts. The heart of the system is varvis itself. This is also where we have the user interface, where the user interacts with the data, can configure virtual panels, configure HPO terms for patients, classify the variants in a very standardized way. It's also where you have access to the variant and CNV database. And we have 2 other components that feed the data into varvis. This is on the left side of the screen, varfeed. This is our bioinformatics pipeline, almost like automated. This is where the CNV, SNVs, and indels are called. It's also where the QC reports are generated, and this is also where we perform validation. And on the other side, you see Alexis, which is our reference data network, where our customers basically submit the variant they have detected, including the classification and the phenotypic description, to the database, and this database can then be queried from all users. And yeah, it's also a nice way, because we manage like variant annotation sources centrally within Alexis. It's basically possible to re-annotate also old cases against Alexis, so that you can also look at older cases that have been looked at last year, and you can re-annotate them so that you have like the full up-to-date data available for historic data. The variant calling pipeline works basically as follows. So this is what happens in the wet lab at our users' labs. So the raw data is generated. Usually then FASTQ or BCL files are generated, and this is then uploaded to our systems, we perform alignment, both Wheeler aligners, a standard algorithm, and then variant calling is performed. So we have basically SNV and indel pipeline and the CNV analysis in parallel. And as I said before, it's possible to choose like the germline automatic pipeline. This has to be configured at the beginning and then can be used. And variants that are detected accordingly, they show up in varvis and are fully annotated against all available information. So when the data is in varvis, the standardized workflow looks a bit like this. So we have the sequencer and the CNV and SNV analysis that are performed basically in our system. So this is not your task in this moment anymore. So it's been outsourced to varvis. The next step would be to connect the analysis information with the patient information. So you can configure like family relations, configure phenotype descriptions, and so on. You can then perform a quality control, including checking that the coverage is sufficient. Then CNV and SNV filtering can be performed. For this, you can use all type of different or kind of different annotations, including literature, also including ClinVar, NOMAD, and so on. And the variants that are classified then according to ACMG can then be transferred to reporting. So for all these steps, when you have like a LIMS system in-house, it's possible to use our API to transfer information automatically between your in-house LIMS system and varvis. And this step, the CNV and SNV filtering, is can be the most challenging step. So where you would need a lot of features to help you to save some time. And so I would like to hand over to Irene. She will show you in the software what kind of features we're offering, especially for whole exome cases. So I'm introducing to you Irene Patric. She has a master's in biotechnology and many years of experience in the field of genetics and research, and also in clinical settings. And she's joined Limbus almost 2 years ago, and she's product manager and clinical application specialist. So I will hand over to Irene for the varvis software demo.
Thank you, Yvonne. Like Yvonne said, I will quickly take you through varvis. I will show you a few cases that I've prepared so that you are able to understand how the tool works, and I will also give you an overview of the structure of the tool. So let's start. And now this is the varvis user interface that the users normally work with. And this view, in the person's view, we have the list of cases, including the cases that have been previously analyzed and closed. So any person can be searched here by entering, and then it displays in the grid with the different columns about each individual case, including which enrichment kit has been used for the particular case, and if there are any reported variants, and the status of the final report, and so on. And then we have the classification alerts, where if you have reported a variant in the past, and then if there is a change in the classification, you have this little badge here which appears saying that there are changes to these many, and then maybe you can decide to look into it and maybe also revisit the case if required. Then is the search view for research purposes, maybe, where you can search for a particular gene based on a particular gene, if you'd like to filter results, or for a variant or a disease. So this can be done from this view. And the sequencing runs is the QC module, which is available for review. And the QC metrics can be reviewed here, either for the whole run or also for individual samples. And I will also show you a little bit. We will have a discussion about this, and possibly Lena will give you more details about this during her talk. And then we have the configuration, where we are able to create virtual panels. And when the user creates panels here, these panels are made available across all cases that the user will be using later. For example, if you would like to create a secondary gene list, ACMG gene list as a virtual panel, and then this is made available perpetually, and you can apply this panel for any case that you are working on. So now I'll come back to this view where I have selected some cases, some previously worked on cases, so that we can go through them one by one. So I have highlighted them here. And as the first case, I would like to take this one, marked as sample 10. And this is the button that I use for opening the case. And as soon as you enter the case, this is the view that we would come across. You see the individuals in the case and their affection status. So in this case, it is the index affected and the parents are unaffected. And you'll also see the different analysis over here linked to every individual. And here, if you see, you find something called the annotation status. And when you click on this, you will be able to see which version of annotation has been used when the case was first analyzed and what is now available. For example, you can see that there is a different version currently available for ClinVar. And this is the information that would also be displayed when you are revisiting a closed case, possibly after a year, because you have to reanalyze and so on. And you would be able to see that if there is a difference, the user can choose to update annotation so that you enter the case with the updated data in place. How is this useful? So if there are new annotations available, new variants that have been classified as pathogenic or likely pathogenic, then it's very useful to update and then proceed with the case. And then over here, you see that this is the virtual panel I spoke about a little bit earlier. For now, we would proceed with all genes in the case, but you could also choose to have a virtual panel. Now, coming to the information about the case, you can click on the person information here, and we can see that this is a male with neuro features like neurodegeneration, leukodystrophy, and breathing dysregulation. So it's also possible that as a first step that I can choose to apply a leukodystrophy panel, but for the sake of the demonstration, I'm just going ahead with all genes. And now I would like to take you to the variant grid right away. And as you can see, what it's loading is more than 80,000 variants. And these are variants that are called out from the index, the mother, and the father. So you can see that there are certain rows that are only present in the parents and absent in the index. Here you can see that the whole grid content can be downloaded. I will come back to the report basket a little later. And these columns are configurable. Like you can move the columns, for example, this way, and you can resize the columns. You can set what columns you want to see, and then also save the configuration of these columns. And if you see here, we have a column menu where there are a huge number of columns available, but I have only selected the ones that I want to see, so that also the loading of cases are faster. And I have also set that I want to see a particular configuration for this case setting. For example, I have a special setting for trio, and I have a special setting for carrier setting, and so on. And I apply this, and automatically this is what happens. Now, these configurations can also be saved for oneself as the default particular user, or also can be shared across colleagues in the laboratory. So you can decide that as a laboratory, this is what it's better that all of us see this at least minimum, and then shared configuration is possible. And the same is with these filter buttons. So now these filter buttons can be created by the user by simply going to this create filter and applying certain filters that are required, and then choosing what we need. For example, here I've just, as an example, filtered for AD, and then come to this filter button and say, I want to save this filter as an autosomal dominant MOI filter. And then when I save it, it automatically appears here as applied. So creation of filter buttons based on the information in the columns is very easy, and it can be switched on and off with just a click. And then on the right-hand side, you have the inheritance filters, which are based on the genotypes of the individuals in the family. Now, I can now show you how it works with these filter buttons. As a start, we would like to apply a filter for known pathogenic variants. Now, this filter button has been configured such that variants that are known in ClinVar and previously reported in Alexis, that is by either the laboratory that is working on this or by the other laboratories. So now anything reported previously will be included here. So this is the button that I will click, and all known variants are being here. And then I say I want to look at variants that are pathogenic, that are present in the index and not other family members. So then this is just an additional button. And you already see that the number of variants have reduced. Then coming to how we can go about this further, there is a column called HPO similarity score here. Now this is this score is calculated based on something that we developed in-house, not just a one-on-one match of what has the HPO terms entered for the gene versus the HPO terms of the HPO terms of the patient versus the gene, but rather it takes into consideration the specificity of the term and also the hierarchy of the term in from HPO, and therefore each variant is assigned a particular score this way, which we can choose to sort, and then you already see that it is sorted based on the score, and the ones that are highly relevant come on top. Now, as we can start looking at this list from the top, and when we click on every variant, the variant details appear here, but also we can see the segregation pattern here, and based on this I can start including or excluding, and you can see that this variant is our variant of interest. Now, when I click on this variant, like I said, the variant window gives us all the different details, and I will just open this further to show you how it looks here. Everything including the coordinates, the genotypes of the different family members appear here, and the significance of every variant that has been previously reported in ClinVar, in Alexis, and in the in-house database that is specific to the laboratory is seen here. And then there is also the overlap information that appears here. And then we have information from the different databases, including NOMAD and the bioinformatics scores. So all this information can be reviewed from this window. Now, if we like to see how this variant looks in IGV, all we need to do is just click on the coordinate, and then we already have this particular variant opening in the IGV so that we can review how the reads look. Coming back here, once we say that this is our variant of interest, the variant can be classified right here in this window, which is a variant classification window. So all I have to do is select the criteria that is applicable for this variant. And as I mouse over every criteria, the tooltip gives what the criteria is linked to, and I'm able to select, and you can see that the classification changes as I am selecting the criteria, and I can add comments if I'd like to, and then confirm the classification. Once I confirm the classification, this will also be reflected here to say that I have classified one variant, and this number is reflecting here also. And if I choose to report it on the default transcript, I can let it be as it is, or if I would like to report it on a different transcript, I can also choose a different transcript from this window for reporting. And then I can go to the literature window to see if I would like to select any particular article that I would link for reporting purposes. And I can apply, you can see that there are different columns here, and I can apply further filters like I would like to be specific for the variant, for example, and then I say these are the literatures that I wanted selected for reporting, and then these would be also available in the report view. Now, coming back to this, now this is not always the case that I apply a filter for the pathogenic and then it's already there in the first view. So in the otherwise, I will have to just go through the whole case with a regular filtering procedure. And for that, I would apply these other filters that I have configured, and I will just take you through what these are now. One is having a default quality pass criteria set. The next is filtering out benign and likely benign variants from ClinVar, which is these are filters that are normally applied for a regular whole exome case. And then I would like to filter for impact, with high and moderate impact, and also the frequency. And then as you can see, this is already sorted for the HPO similarity score. And then I can start applying the inheritance filters in addition. And like I said before, these are based on genotypes. So the de novo filter is going to look at what is present in the index and absent in the mother and father. And you can see that there are several variants that do not have an overlap and those that have a poor overlap. So, but just by looking at a score, I already get a hint that these are not relevant enough. So then I can just continue to proceed to the other filters. And you can already see that the variant of interest is appearing on top because of the HPO similarity scores. And this way, we can continue to apply the other inheritance filters. And as you can see, there are also some filters that are disabled based on the scenarios. So basically, this is also linked to whether the affection status of the different family members in the case. So now I can show you how the report view looks. I will go to the report view, and then the one variant that I had classified is also appearing in the report view, and I can choose to enter the disease, the variant category for which I'm reporting, the mode of inheritance, genotype, clinical assessment, and then save this information and send it for a second level review, or I could also approve and complete it right away such that I can download the report from this view. And after this, I can mark the report as solved or partially solved so that I can also revisit such cases later if required. Now going back and moving on to the next case, I would like to show you another neuro case as well. And this case has nystagmus, ID, seizures, including cerebral atrophy. And this is also a trio. However, in this case, I choose to apply a panel for morbid genes. Now, what morbid genes panel is it has genes that are have an entry in OMIM or genes that have HPO term tagged to it. So it's just shortlisting this to clinically relevant genes, basically. So I am going ahead with this panel for the trio, and I will again move to the variant view. In this case, again, like I mentioned earlier, you can see lesser number of genes for the trio. It's because everything in the case is restricted to the genes in the morbid genes panel. And so once you apply a panel in the start of the case, everything after this would be restricted to the genes in the list of the panel. And here you have now the same variant grid with the configurations. And as a first step, as in the previous case, I also apply the filter, and then I sort it with the HPO similarity score. And you already see that two variants of interest. So these are variants that were, these are cases that were previously solved. So I have highlighted these variants so that it's easier to identify. And we have also classified it as likely pathogenic variants. So you can see that just with a few clicks, we already have our answers in this list on top. But now I would like to show you a little about the report basket. When I apply certain filters, and I would also like to save the steps of filtering. So this is why the report basket is there, that now I say whatever I see in this view, I would like to save it to the report basket so that I know what I applied, what filters I applied, and what is the result I saw, and how I went about my filtering steps. So that at a later time point, when I have to revisit this case, either it could be either me or a colleague of mine who would like to know how I went about this case, it would be easier to see what was applied and what the results were at that point of time. So as I mentioned earlier, also with annotations, there could be changes with the data as such, with the classification and so on. So it would always make it easier if there is some way to see the history of users. So this makes it quite easy. And also when you have nothing, it's also easy to say that you had nothing when you applied such and such filters. So this, when we save the view, it goes to the report basket and also gets saved as an attachment, which I will show you a little later. Now, as in the other case, I can continue to apply the other filters. And go ahead with the same filters, and we can see the number of variants that are appearing for every filter this way. And we can also apply further filters to the columns as we are already working on set filters. It's also possible that I can apply further filters. For example, when I'm looking at CH, maybe I choose to apply something like AR in addition to what I have. And then I'm restricting my results further. So this is completely flexible and up to the users how they would like to use it according to your laboratory SOPs and so on. And it's quite easy to also share such configurations. Now, coming to the report view, like I said earlier, everything over here can be approved and completed. And then this information of the report can be downloaded. The whole information over here can be downloaded as a final report. And what was in the report basket can also be downloaded as an attachment. Now, I would like to show you.
Irene, Just
Yes.
before you switch to another case, there are some questions that have been already
Sure.
raised. So one question was, can the CNV and SNV pipelines be interrogated together to identify compound heterozygous situations?
Yes. So right now, we have it in such a way that when we are looking at the CNV, we have I will be able to show you a little later a track for the SNVs as well, where we can see them together. But we are currently working on displaying this together in the SNV grid. When you have a compound heterozygous situation, soon we will also have the combination of CNV plus SNV in the grid itself.
And soon means October.
Yes.
Yeah. So it's not like it's actually already done.
The final testing phase.
Yes.
The end of October,
testing.
this is available.
Yes.
Yeah. Another question is, can varvis suggest an ACMG or AMP classification? So you just have to check the criteria, or do you always have to select the criteria yourself?
Lena, would you like to take it?
Yeah, I can. So right now you have to select the criteria yourself. We don't do a classification. There's a specific reason for that, because we are a medical device, and that we are bound to extensive validations, and also to make it possible for the user to absolutely transparent how the software actually got to this result. For that, we have actually a research project together with the University of Rostock. So we are working on this right now, but it will take a little bit more time. And then we will also have a pre-selection like this, but at the moment not, because we have to be extra careful and need other precautions that other software companies don't need to take.
Okay, there's one more question regarding ACMG classification. Can you change the strength for ACMG different criteria when classifying variants?
Yes. So if you had a look at the variant that I just showed, I can open this again and maybe. So the change in strength is possible. So it's it's we have provided that option, that when you select the criteria, you can also see what you want to apply together with it.
And this would also be stored in the
Yes.
in the rational, basically, right? That is stored with the classification.
Yes.
Yeah. And do you apply specific rules developed by clinician expert panels for classifying variants in certain genes? Is another question.
Should I answer?
Yeah.
Sorry. So right now we cannot. We had it actually prepared, and so it would be actually for us, if it's desired, we could implement it pretty easily. So the whole ACMG setup, you see, the whole calculator is designed in a way that we can easily change it and adjust it to any kinds of updates that are coming, because there are frequent updates. So right now we don't have it, but it would be something we could do until the end of the year. Yeah,
this is also reflects in the header of the panel, right? ACMG version 3. You see that, you know, new information is incorporated and reflected in the new version, also for different types of variants. And can you add a comment on the criteria? It's also asked.
Of course, yes.
So you can just add comment easily. Exactly. Okay, let me check. There are a lot of questions. If we answered them all.
Oh, the coverage information.
There's one regarding the coverage, but maybe we can address this when we open the next case where we have like a small targeted panel. So can you access information on the coverage for each exon of the whole list of genes of interest? So of the virtual panel, you can show this in the next case that we will take a look at.
There's actually a follow-up regarding the comment. I think so. I think it's asked if you can comment on the criteria in general. We don't offer that you can comment on any ACMG criteria, but we have a lot of comment functions. So you can comment on variants, you can comment on the gene, on a gene region, basically. And but not on the criteria itself, because we actually stick to the ACMG description here.
And the comments are not only user specific, but it is something that is shared across everybody in the laboratory, right? So it wouldn't be a problem if you also commented on the variant, because it's anyway visible to everybody.
And then there's a question, can you curate variants in different contexts? Germline versus somatic. I'm not really sure what this is referring to. I mean, we do have a somatic bioinformatics pipeline and a germline pipeline. So of course you can curate them differently, I suppose. We don't have, if it's referring to the ACMG criteria, we don't have specific somatic ACMG criteria yet. If they are available, this would be something we could consider implementing.
Okay. So I think we've answered these questions,
so
we can move to the
next case,
last case here. So I would like to show you mainly the CNV, how it works over here. So I've chosen a simple case so that we can discuss some aspects and make it clear. Now this is a breast carcinoma case, and therefore we have applied the HBOC panel. And of course, we have both the SNV and CNV included, but before I go to the CNV view, I could show you this request about coverage. So basically, when we look at the what the panel entails, you can quickly have a look from here what genes are included. So because only these genes are going to be applied for all the following steps, including the coverage and the CNV, the variants. So these are the genes that are included. And when we want to make an edit to the virtual panel, it is possible also from here, but any changes made here will only be a temporary fix specific to this patient. And if you want to make a permanent change, then it is from the other window where you are creating the virtual panels. So any change can be made also on the fly from this view, but also the information is restricted to these 5 genes henceforth. So the coverage information is also available for these genes. Like you say, so for example, over here, the 20x coverage is 100%, that you can see that is for all the genes. So then I wouldn't have to be concerned about finding any deletions within the case, basically. And then also you have exon level coverage information available for every gene in the panel. So I think with this, I will move to the other.
Yeah, we have some questions also coming in. So I'm sorry that there was something wrong with this case, but one question is, how do you select which transcript to use for annotating variants and coverage analysis?
So this is transcript basically selection. So we have a whole course about transcripts selection, and it's available on our varvis Academy. So I think this course is also available for guest users. So we would request you to take a look there. where we have detailed many things about also why alternate transcripts are important and so on.
And we can add that we can basically configure this as you wish, right? If you want to have as the RefSec select transcript as your preferred selected transcript, you just need to indicate this, and then we select this automatically. If you have a specific list, a specific set of transcripts you want to use, this can also be provided and be configured accordingly. So this is configurable. And we have questions regarding which minimum and maximum sizes of CNVs are detected.
Yeah, so the aim of the case was that I would be able to show you that we are able to detect even single exon deletion. So that case had a single exon deletion. Of course, this requires that the case has very good QC metrics. You know, it requires that you have good coverage, good depth to be able to go to that level. But of course, we have also had cases of partial deletions and so on that we were able to detect. So it is possible when the QC parameters are satisfactory.
And there is no maximum, right?
There's no maximum. Yeah.
Yeah, the limit. I mean, the chromosome would be the maximum limit.
Does the coverage information apply only to the coding sequence or also to adjacent intronic sequence?
Yeah, so
do you want to take this?
Yeah, so you can actually you can choose. You can either say I want to restrict it to the coding sequence, or you can not do that and also enter a certain kind of padding that you can decide on how much information into the intronic sequences you want to show. Also, it is possible if intronic regions are included in the BD file for the analysis to see the coverage of those if they are far outside of any coding sequence, for example.
Yeah, regarding the maximum and minimum sizes of CNVs, we also have in the next couple of slides, there is one section about validation of CNV. So this will also be readdressed again.
So I will give you some overview into what varvis Academy is, the support system and the onboarding services that we offer to users. Now, we've tried to keep onboarding as simple and straightforward so that it's not a very difficult process, but quite easy to go ahead, for which the starting point would be a free software trial. And we have no obligations here, and we are able to guide you with a setup for clinical routine where the users will be able to test clinical cases in their trial instance. And then once you are a customer, we are also able to help with validation, which I will be detailing a little about later. And then we offer training. And then we also have full access, we give you full access to the varvis Academy. And then, of course, we are there to support you throughout the process of setting up and also later when problems arise with the cases that you have either as technically also on at some technical aspects we are able to support. Now, the validation process is quite an important step. And for this, normally a reference sample that contains a known set of variants is sequenced and analyzed, and then the variants determined from this data set is compared to the reference data set to see how the process is working. And for this, why is this important is because we need to assess the performance of the workflow, and so that we know how sensitive and precise the assay is. Now, varvis as a software has been validated and proven in a clinical setting, but how would it perform in combination with the laboratory processes, right? Because laboratory processes influence the QC parameters a lot. And so together we must see how we are able to attain a particular sensitivity, precision, and so on. So this process becomes then important to be done together. So I will tell you a little bit about our pipelines that were validated. Now, if we talk about validating using a reference samples, now what would be a good reference sample? Genome in a bottle samples, which has been globally recognized and recommended to be used as an official reference material. Now, these samples have been characterized very well by multiple methods. And so there are a known set of variants, and there we have a known data set, basically. And then when we are able to compare the sensitivity and precision based on the data that we are generating for the current workflow. So for most cases, for a WES, we are able to see greater than 98% sensitivity and greater than 97% precision. Now, there are certain sensitivity requirements that are set up as the guidelines for medical standards, and we always ensure that any change in the tool or the workflow, we make sure that we are in compliance with these requirements. And so for what does this mean for the laboratory? What would the laboratory have to do? Just acquire this reference sample and sequence it in the laboratory so that we take care of the variant calling, perform the annotation, and then give a validation report. Basically, this validation report will be given back to the users so that even they can use it for their accreditation. And this whole service of validation we provide at no cost. Now, our CNV pipeline has also been validated using thousands of samples from routine diagnostics, and also the results were compared with PCR-based methods. Now, what we saw was 100% sensitivity when compared to PCR-based methods, and a greater than 95% specificity. Now, what is the benefit of validating this pipeline? Is when we have when we're looking at CNV from NGS data, and if the QC parameters are not okay, then we definitely have to rely on other methods. But if it is okay, and when you have no CNV calls, then you are able to generate a negative report with confidence. Now, the advantages of validation and automation is that we're able to trust the results because the whole thing is compliant with all the standards that are set. We have a reduced TAT because everything is available together, including the CNV analysis. And of course, we have an increase in confidence, and there is a huge time and cost saving because we don't switch between tools and everything comes together in varvis. Now, varvis Academy, which I mentioned earlier, is the training platform that we have where the users, everybody from the new users to existing users, are able to benefit from this by revisiting the material that we have over there. We have courses for guest users, like the one that I mentioned, the transcripts, but we also have a course for carrier screening, for example. This one gives us the tiered approach to of ACMG, the recommended ACMG tiered approach is explained here very well. And then we have a course about pseudogenes, which explains why this becomes important in the context of NGS analysis and how alternate transcripts impact analysis and so on. So there are some courses available for guest users, but of course, the whole access is available for the varvis users. And in the whole access, you will see Courses like the West training course, and which sort of gives a glimpse into the regular practices of West's analysis, and the basics training course, which has certain features of varvis explained in detail. So the users are able to get in-depth knowledge in certain topics. And we have courses also for advanced users. So when we have updates on our software, we always make it a point that the users understand what updates are, and what improvements were made, and how it affects the workflow, and so on. And of course, any information or anything that is made that is useful for the users is always also made available on the platform, so that they can revisit anytime and use the information. This is just an example that I have given here of how the course looks like. The courses have sections, as you can see over here, and several real-time cases have been explained. And as you can see here is also a CNV case that is being explained in this video. Of course, this has audio, but right now it doesn't, because I'm just playing it for the sake of demo. And so it's more like a video where you will listen in and also observe the case, how the case is going about for analysis. And in this case, the CNV analysis is being explained. And then you would have many interesting quizzes as you're going through these courses. And of course, when you need a certification, that is also possible by giving a test and answering some simple questions based on what you understood from the course. And if we can go ahead. Yeah, so that was about how the courses look in the academy. And of course, support is a big part of our service, that we have a group of experts, I would say, who take care of everything from smooth onboarding to answering complex case-related questions. Because our team also has expertise in laboratory processes, we are able to guide the labs with improving their sequence quality by giving feedback and so on. So everything from onboarding, giving QC feedback, and solving several case conflicts, and solving complex questions about SNVs, CNVs, and several other questions that also Lena will be showing in the following talk, will be better explained by her. So you will see that our support goes a long way along with the product. So with this, I will hand it over to Yvonne.
Yes, thank you. There's one question coming in. Let me briefly read it. Is it possible to compare whole exome sequencing from multiple patients in order to identify causative genes or variants for a certain disease?
For multiple patients, so basically I assume you're talking about multiple individuals who are affected in the same family, or if you're talking about large cohorts,
Yeah, I think it's more a research-based question, as I understand it.
Lena, would you like to take this?
Yeah, so at the moment, large cohorts would not be possible. So you can have, right now, you can have families of up to 10 family members.
Also
yeah,
more.
so, but we are working on, because there is a database, a large database. You will have, each customer has their own big database in the background of the cases they have analyzed already. And additionally, of course, there's an Alexis database, which summarizes the results of all our customers. And so right now we're working on a tool to give access to this database, which would allow exactly this kind of thing. Like, okay, give me all patients with, of course, these patients would be only restricted to your own local database, but give me all patients with a certain HBO term our symptoms and then show me their variants or something like this. So this is something we are developing right now. It was not originally in our idea because we said we are going for clinical diagnostic labs, and this is more research topic, but a lot of our customers also do research. So this has come up quite often, actually.
Yeah.
Yeah. So this is
there's an overlap between
feature.
research and clinical diagnostics. Yeah, so as Lena has already taken over, still I want to take the chance to briefly introduce her. So Lena is a PhD in molecular biology and like long years of experience in like a clinical laboratory where she served first as a head of duplication and deletion, and then also as a senior director for genotyping, where she was responsible for the entire lab process and workflow optimization. And since 2018, she's with Limbus, and now she's the head of product management and service. She's basically one of the key links between the customers, the wishes of the customers, and the software development. And she's dealing with a lot of questions and requests. And here in the next talk, you can basically see kind of a best of cases they've been reviewing in the support team. So the title is Cabinet of Curiosities, Detecting and Interpreting Extraordinary Variants with varvis. So this is not about the easy cases, but the tricky ones.
Yeah, exactly. So thank you very much. So this basically, since we do a lot of support and we also see a lot of data across different platforms, different enrichment kits, we actually by now have quite an extensive knowledge, and which we like to use to help our customers. And so we have a very close relationship with our customers, and get a lot of answers that are not strictly regarding our software, but which is fine, and we like to answer those as well if we can. And so this talk is basically not about functions, features in our software directly, but more indirectly what you can also see in our software when you do not have to trot the normal ways, but if you find something that's a little bit odd. Yeah. So with that, I would like to jump into the first case. So you will see a lot of IGV, because this is something that we also incorporated in our software. So actually in two scenarios. So one is in the CNV view, we have an IGV incorporated so that you can see the BAM files in the CNVs view, but also if you click on any coordinate at any point in our software, you have the ability to open the BAM file to this case in an external IGV application. And so the cases I will describe will often be only explained if you actually take a look at the BAM file. So this was a normal case. You see, it's a normal BAM file, but what sticks out was these weird spikes. And so the customer asked us, did you do the alignment correctly, basically? And it was a whole genome sequencing sample. The sample was a Bacchus swab. And what was odd about it, there were a lot of de novo mutations in the index, a lot of insertions, all over were the spikes. None of them were detected by our CNV analysis, but the area was very small, but had incredible high coverage, and there were long stretches of mismatches on both sides. So this was pretty easy. What we did was, okay, we said either it's really insertion, but was a little bit unlikely, or a contamination. So we just blasted one of the mismatched reads, and then we found out, okay, it's a bacterial contamination because it's a Bacchus swab with oral mucosa. So this is one thing you have to keep in mind when doing whole genome sequencing without enrichment. If you combine it with Bacchus swab and not a blood sample, you might enrich a lot of bacterial contamination, bacterial DNA, and some of that will align to the human genome. And on the other side, this is also a task on our roadmap to get these removed, which is not that easy because it could be any kind of bacteria. So the next case looks on the first glance looks similar. It's again area of very high coverage, but this time it was a smaller NGS panel. It was a blood sample, and in this case there was an insertion detected by our CNV analysis. The insertion was detected in exon 23 and 24, but the problem was it could not be confirmed by MLPA, by the customer, and so they asked us what happened here. And we did take a look at it, and so there were clear arguments for an insertion. We had clear breakpoints. There was an heterozygous variant inside of it with an allele frequency of 0.3 and 0.7, which was nicely consistent with an duplication, and but it was very small, and it could not be confirmed. So we asked them, so what struck us was there were a lot of duplicate reads. And what we also found very interesting was that there was very clear breakpoints, and those were exactly where the exon started, or a little bit before. And then we asked for the Sanger primers, and then they were exactly at the positions of the Sanger primers. And so the result, and then there was this very variant inside, and we checked the database of the customer, and it was super rare. They had it once before. No one of our other customers had ever seen this variant. And so actually it was a contamination of the NGS library prep with a Sanger PCR product. So they had detected this variant before in another patient a week before, and had done a Sanger confirmation of this. And then for the next patient, they had the contamination then. So this was a quite very nice reminder that the PCR of yesterday is the biggest enemy of the molecular biologist. And the next case was an whole exome sequencing with a blood sample. And this was pretty clear. There was not much confusion. It was an insertion that was also detected like this. It was de novo. It was encompassing 2 genes. But the breakpoint was very clearly visible, which was nice. And there were very big reads with mismatch. And the question of the customer was, is this the real breakpoint? And might it be a tandem duplication? And normally this is questions you couldn't answer with a short read sequencing. So what we did is we blasted the reads and then did take a look and saw that every read, and there was like the area I showed before was the same. That was basically this end, but we had another similar looking area at the other end, at the other gene, and we blasted reads from both sides. And all of them had an upstream end matching to ESCO1, and all of them had a downstream end matching to GREB1L. So the logical conclusion was actually it needs to be a tandem duplication, because otherwise those reads wouldn't match. And yeah, so this was a nice example that when you're lucky, you can even find a tandem duplication. And if you pay attention to the reads and the mismatches in them. The next case is one where we had a gene and all exons had this weird shape, and all the introns seemed to be deleted. So there were a lot of heterozygous deletions of all introns. So it was like, is it heterozygous deletions of all introns or insertions of all exons? Or again, a contamination with a PCR product, because the breakpoints are exactly at the exon and beginning and ending of the exons, or some kind of complex structure variant. So what we did here is we just did take a look at the BAM file actually. So the thing is, you see the typical increase and decrease of coverage, which is typical for hybrid capture assays. And this was a hybrid capture assay. But you see also the steep incline and the exon-intron boundaries, which looks more like amplicon-based. But what you actually see here is 4 copies, 2 copies of the wild-type gene, that would be the hybrid capture assay coverage, and then 2 copies of a processed pseudogene. And for those who don't know what a processed pseudogene is, this is a spliced sequence which are introduced back into the DNA via retrotransposition. And this is also part of our Learn Worlds course that Irene mentioned before. And one of the reasons you should watch that video, because the pseudogenes have a lot of surprises. And so then the next case we have is actually this one, where this is a screenshot from our software where you can see, as you clearly see, there's a duplication of the whole P9 arm. And the problem here was that the customer had done a chromosomal analysis before, but couldn't detect it, didn't detect any triplication. And so this is one of my favorite plots. It's one of the sequencing runs menu that Irene mentioned before, which is a menu where we show a lot of QC data and plots to help you evaluate the data, and also might sometimes give hints of what the patient have, what kind of problem the patient might have. So in this case, you have in the case of this plot, you have on the y-axis the mapped reads, the fraction of mapped reads, and on the x-axis just the chromosomes. And you can clearly see, so every line is one case, and you can clearly see that all samples behave more or less the same, except for one, and this is the one we were looking at before. And you see that this sample has a lot more reads on the chromosome 9 than all the other samples, which supports the triplication. And to further confirm that we were right in this matter, we also did a plot of the allele frequencies, and usually you would expect, like in this green line, you would expect a peak at 0.5 and 1 at 1, 0.5 for the heterozygous variants and 1 at the homozygous variants, and the index in this case had a peak at 0.3 and 1 at 0.7, which is consistent with triplication in this case. Also, we plotted a little bit different as a scatter plot, this time the whole chromosome, and you can clearly see this is a P arm, this is a gap of the between the 2 arms, the centromere, and then you have the Q arm, and you see the Q arm is normal with variants clustering around 0.5 and some variants clustering at 1. Usually it looks a little familiar because you might see this from know this from the arrays. Then you would have another plot down variants here with 0, but since this is data from our exome, we only show variants and no wild type. So there's no you have to imagine the variants, also the normal SNPs down here. And for the Q arm, for the P arm, oh yeah, now I'm confused. For the P arm, you see now that the variants are actually moving apart and cluster around 0.4, 0.6, something like that. So this is easily seen here. And so we went back to the customer and said, okay, we are pretty sure it's a triplication. And they redid the chromosome analysis, and then they could detect it because it's mosaic. And what happened was that they used different type of blood cells for the chromosomal analysis than was predominantly present in the blood sample that was used for the NGS analysis, and therefore they couldn't detect it before. The next case was one where the customer said, oh, I think we mixed up a sample, because what we have in our QC module is a gender check. So we the customer has a possibility to enter the sex of the patient, but additionally, we also calculate the sex. And then we check if those two match. In this case, it didn't match. this is again a screenshot of my favorite plot from the sequencing runs. Now only basically the XY chromosomes. And so what you can see, the green line is a typical male with half amount of reads on the X chromosome, and then compared to a female patient, and some reads on the Y chromosome. So this is typical male. The blue line is a typical female sample with double amount of reads on the X chromosome and nearly no reads on the Y chromosome. And the orange one was the one that was the case in this, and that was XXY. So the sex match might even indicate to you that there's something wrong on a bigger chromosomal aberration area. And the next one is we also provide a contamination score for bigger panels. So everything that's like big clinical exome, whole exome sequencing, or whole genome sequencing. And the contamination score was validated by us in vitro with the help of MGZ and Andreas Lana's team, and with a lot of positive controls, and also in silico with 1,000 cases. And we can now recommend a threshold of 0.3, and with that we are able to detect maternal contamination in prenatal samples with a limit of detection of 1%, which is, of course, so detect maternal contamination in a prenatal sample is a lot more difficult than just detect contamination of one sample with an unrelated other sample. So of course you can detect this also. Yeah, thank you. And so in this case, the contamination score was increased. Also, the head-hom ratio was increased, but we had a little bit trouble. So the customer had a little bit trouble to believe that this was a contamination. And so we did our delta F distribution plots again, and it was clearly shown on the left side. You have a normal patient which clustered 0.5 and 1. And here you can see that the homozygous variants are actually smearing a little bit downward. And so we were able to detect that this is a contamination of 12% with some other patient. And we determined the 12% by looking at the CNV, because luckily there was a CNV in this area, and by the log2 value, and the value was not exactly how it was supposed to be for a heterozygous deletion. We could calculate how much contamination we actually had in this case. And so in the end, they actually redid the analysis, and then the contamination was gone. Yeah, and that was basically already my last case. Thank you very much for your attention. And in case there are any questions, I'm free for any questions. And
so far,
control back to you.
Yeah, so far there are no questions. I would still, except for the positive feedback from Andreas Lana, which is very nice. I would like to invite you to stay in this session for another couple of minutes to ask, have like an open discussion if you are interested to ask questions. I still would like to, if I could jump to the next slide, let me try again. Yeah, for more information, you're invited to visit our website or also contact us directly. If you have questions regarding this talk or the software itself, if you're interested in a demo for your lab, or also to have like your own varvis software trial, where you can, like Irene already showed you how this would work, so you can upload up to 80 samples and then have like a training, and then like a 2-week period to test the software with your own data. So this is a possibility that we provide like without any obligation. And yeah, feel free to contact us if you want to if you want to know more. There's one more question now. Kindly, interesting cases. Kindly may you clarify the case of amplicon and capture-based case?
Yeah, okay, so okay, I tried to explain it again. What happened is, so there are process pseudogenes. So these are
You want to have
the
the slides again?
should I? Can I?
I would
You want to?
hand over the control.
Okay, so the process pseudogenes are pseudogenes which have already undergone splicing. So the only thing that is left are the exons. So it's like, so basically cDNA. And then this cDNA got somehow put back into the genome. And of course, our short reads also align there, but they will only align at the exons because the introns are already gone. So you basically have for, I'm just checking if I can go back. Yeah. So you basically have four copies. 2 copies of the normal wild type gene. This is unfortunately a little bit slow, and I don't have the, yeah, this one. So, so you have basically 4 copies. You have 2 copies of the normal gene, including exon and introns. That is what you see here. And so you basically underlying, you see here where there's no exon, you have normal coverage like you would. So normally the curve would be going like this somewhere. Oh, you know, something like this. Or like this, depending on where the probes are. But there are also reads that were aligning to the pseudogenes only at the exon, and those are basically on top. And since they were spliced, they start immediately with the exon-intron boundaries. So every time you see something where you see like you see like there are steep inclines or at exon intron boundaries, you have to think about a pseudogene, especially if the whole gene looks like this or a big part of the gene. So in this case, the whole gene looked like this. Every exon had this. And there are also, we already had cases where you didn't have this underlying wild type gene. So in this case, you would have no intron coverage and it would just start like, it would clearly look like an amplicon-based assay. It's just an example for me because so it's not an amplicon-based assay. It just looks like one, you know. So I hope that made it a little bit more clearer to you. I think that's a question for you.
Thanks for the clarification. So I think it's clear now. Yeah, there's one question: how much is the software after the onboarding process? I assume this is a question regarding price. So the pricing model is per sample, per usage. So you basically pay really for the samples we process for you, and all other services like the usage of the software, the annotation, CNV analysis, onboarding, support, validation. This comes in a full package, and the price depends on the size of the sample or the size of the panels, and the annual volume you're committing to. But as a rule of thumb, so the list prices start between 50 and 75 euros per sample for panels and exomes. So this is the range you can expect. But genomes are more expensive.
Also, there's a lot of included in the price. So it's just a per sample price. You don't have any additional costs. So it includes validation, setting up, support. You don't have to pay extra for fast report, by the way. So we will answer within 1 day, regardless of how much you pay. And yeah, so there are no additional costs unless you want long time storage. In that case, you have to pay basically the storage. We just give the prices for the storage of the cloud. Yeah. But that's it.
And one more question: is your software mirrored in Asia Pacific region cloud or only in EU? So our servers are located in the EU. However, access can be provided from all around the globe, basically. So we need to basically, because it's a medical device, we need to make sure to register varvis also in the respective countries. But there's no restriction in terms of technology that it should be available from all around the world. How long are cases stored by default?
So the cases itself is stored as long as you are our customer. Another question is the BAM files. So you can book with us long-term storage, then we will store it for you in the cloud. And then you have access to the BAM files as well, as long as you need. Otherwise, I think the BAM files are removed after 3 months, maybe.
This can be also negotiated and configured according to the needs of the customer. I
think right now the default is 3 months, but yeah, we can also extend that.
But most customers decide to choose our archive service, where we really also store like the raw data, the BAM files. The advantage of the storage is also that the coverage information is always also visible or accessible from varvis. So even if you process like samples that have been processed a year ago and you open them again and you open IGV, you will still be able to load the BAM files and to see the reads again. If we delete the BAM files from our system, this is basically the link that you will lose. This is basically the consequence.
Yeah, but the cases itself you can look at as long as you have varvis, and you always can also, one important question, some of frequent question is, you can always re-annotate this case without additional cost.
Yeah.
Even after 3 years, 4 years, 5 years, we don't care. It's your data.
Only the processing basically that initiates pricing. Okay, any other questions? Doesn't seem to be the case. If questions pop up later, please feel free to reach out. Also, if you're interested to sign up for the academy, to try for yourself, to be the guest user, to see the first courses, and just get in touch and we can show you the way. I thank you all for your attention, and enjoy the VPTC courses today and still tomorrow. And goodbye for now.
Goodbye.